2026, let's see what the latest research is telling us. Stem cell patches restored heart function in 12 of 20 patients with severe heart failure. A mouse study mapped two distinct lymphatic routes for clearing amyloid from the brain. One fast surface pathway runs along the skull and meninges. The second route, through brain parenchyma, operates more than 30 times slower via diffusion. Researchers tracked clearance kinetics under varied physiological states in the mouse model. The work points to future studies on how these routes shift with aging or disease. Now shifting to a very different area of research. Stem cell patches derived from pluripotent cells were sewn on to the ventricles of patients whose hearts could no longer pump adequately. In the NEJM study, 12 of the 20 participants showed measurable gains in ejection fraction and reported better daily function 6 months later. The patches integrate as living muscle rather than acting as a mechanical pump. The approach offers a potential bridge or alternative to ventricular assist devices. Long-term immunosuppression requirements remain an open question for the next trials. From restoring heart muscle to simpler, non-pharmaceutical ways of supporting cardiovascular health. A systematic assessment examined free interventions such as time in parks, forest walks, and community gardening. The review compiled evidence across physiological and psychological outcomes. Participants showed measurable gains in cardiovascular markers, stress hormones, and cognitive performance. These approaches require no pharmaceutical input and can be scaled at low cost. Follow-up work will test dose-response relationships across different environments. From broad environmental effects on health to a low-cost diagnostic that targets a specific infectious disease. A $4 tongue swab detects active tuberculosis in 30 minutes. The test targets bacterial DNA directly from tongue epithelial cells rather than sputum. It performs well in people who cannot produce phlegm. Validation showed reliable detection of active disease without specialized laboratory equipment. Larger field trials are underway in high-burden regions. From practical diagnostics in resource-limited settings to overlooked museum specimens that reshape evolutionary timelines. A specimen from the Fruoangian period was re-examined in BMC biology. The fossil dates to roughly 497 to 485 million years ago, and reveals greater diversity than previously recognized. Detailed morphological comparison links the specimen to transitional forms between earlier and later faunas. The study narrows the Fruoangian gap with new character data. Additional museum holdings are now being screened for similar overlooked material. From deep time diversity to contemporary social structure research in long-lived mammal populations. Data from nearly 500 spotted hyenas tracked over 28 years showed that different rank calculations predict different outcomes. Some metrics best forecast litter size, while others better predict offspring survival. The Leibniz AVW team demonstrated that a single universal rank score misses these trait-specific patterns. The work highlights why hierarchy studies need outcome-matched variables. Future analyses will apply the same approach to other social mammals. From animal social metrics to human intergenerational health effects. Offspring of 9/11 responders exhibited higher rates of anxiety and behavioral issues, even without direct exposure. The study documents intergenerational transmission through parental stress physiology, rather than shared trauma. Data were collected from families of first responders with documented exposure levels. Researchers controlled for socioeconomic factors and subsequent life events. Longer-term follow-up will track whether symptoms persist into adulthood. From human cohort findings to an organoid platform that models nerve repair, a lab-grown brain spinal cord model combines cortical and spinal tissues to study axonal injury responses. Researchers observed regrowth after lesions previously considered irreversible in simple mortals. Live imaging tracked axon extension and synapse reformation over weeks. The platform allows direct testing of molecular interventions on humanin-derived cells. Planned experiments will introduce patient-specific genetic backgrounds, from tissue models to island populations evolving similar traits through separate genetic paths. Subspecies of wrens on separate Scottish islands increased body size through distinct genetic roots. The University of Birmingham team compared morphology and genomics across mainland and island populations. Strong evidence for island gigantism appeared in 2 isolated groups. The findings illustrate how similar environments can produce convergent traits via different mechanisms. Further sequencing will map the underlying loci. From evolutionary mechanisms to coastal restoration techniques that rely on simple structures. Sedimentation fields using brushwood fences trapped sediment and allowed marsh plants to establish where other methods failed. Sites with intact fences retained higher elevation gains than those with damaged sections. The study quantified a 46% global loss of salt marsh and tested repair thresholds. Maintenance schedules emerged as the key variable for long-term stability. Coastal managers are now testing scaled versions in additional estuaries. From field-based restoration to agricultural nutrient trials, Arkansas field trials confirmed that adequate potassium supply increases both total lint weight and fiber length in cotton. Deficiency reduced boll retention and weakened fiber strength. The work quantified optimal tissue concentrations at key growth stages. Extension recommendations now include tissue testing thresholds for growers. Additional trials will examine interactions with drought stress. Now, here's something most people get wrong about how the brain clears its own waste. Most people assume the brain lacks any equivalent to the body's lymphatic drainage system and must rely on slow diffusional 1. In reality, a dedicated network of perivascular channels, the lymphatic system, actively pumps cerebrospinal fluid through brain tissue to flush metabolic byproducts while you sleep. Right now, as you sit listening, that fluid is moving along the surfaces of your blood vessels, collecting proteins such as beta amyloid before routing them outward. One pathway hugs the skull and meninges and clears material quickly. A second, deeper route through brain parenchyma works more than 30 times slower. The practical takeaway is that consistent sleep timing matters more than most people realize, because the system's flow increases sharply during deep sleep stages. Watch for upcoming studies that test whether common medications or blood pressure patterns alter these clearancer rates in humans. Before we go, keep an eye on durability data from the next stem cell heart patch trials expected later this year. That's Planetterrian Daily for today. If you enjoyed this, a rating or review on Apple Podcasts or Spotify really helps new listeners find the show. I'm Patrick in Vancouver. Thanks for listening, and I'll see you tomorrow. This episode used AI Voice's synthesis of my voice. Editorial selection and analysis are my own.